Hi, my name is Bo Longquist, and today I'm going to be quickly presenting to you about heat shock factor 1, which is a transcription factor and DNA binding protein found in most living organisms. All right, so first I'm going to talk a little bit about its structure. On the side of the N terminal, we have the DNA binding domain of the form helix turn helix, which is able to recognize the DNA sequence NGAAN as its binding site. The oligomerization domains are up next, and they're a little bit downstream. There's actually two of them, both of the form leucine zipper. One is found in between the DNA binding domain and the regulatory domain, and the second one is in between the regulatory domain and the transactivation domains. The regulatory domain is responsible for regulating the transactivation domains in the absence of stressors, and the transactivation domains are responsible for facilitating transcription once the protein is actually bound to DNA. Okay, and I've touched on this a little bit already, but the function of heat shock factor 1 is to actually regulate the heat shock response within cells. This response occurs not just in the presence of heat, but anything from a lack of glucose, maybe it's toxins are present in the cell environment. Anything that induces stress in a cell, heat shock factor 1 is going to be activated and begin producing heat shock proteins. So I'm going to talk a little bit about how that occurs. Heat shock factor 1 is always present in a cell, however, it is not always active. In the presence of stress, however, it forms a trimer with other heat shock factor 1 proteins, and it can then enter the nucleus of the cell where it binds to the DNA and begins transcribing heat shock proteins. These heat shock proteins can then leave the cell and begin aiding in either refolding proteins, or they might chaperone newly synthesized proteins and ensure that they're folded correctly. This is really important in the presence of stressors because proteins are more likely to not fold correctly, and that could result in the cellular functions not being preserved. And this process actually creates a negative feedback loop with heat shock factor 1. As more heat shock proteins are created and there are fewer proteins that need to be refolded, those heat shock proteins will then bind with heat shock factor 1, and it will no longer form as many trimers, which will then slowly begin to inhibit heat shock factor 1 from creating more proteins. And then here are my sources, and I'm out of time, but thank you for your consideration.